Hi everyone, my name is Josh, and today I'm going to be taking you through how to make some CSV style tables and figures in Excel. In the first video, I'm going to be showing you how to do some data processing, statistical analysis, and formatting of a bar graph in Excel. And in the next video, I will show you how to go about making a standard curve properly formatted, and also a table according to CSV guidelines. For more information about figures and tables, I highly recommend a student handbook for writing in biology, which you can find at the Dalhousie bookstore. Okay, so without further ado, we'll hop into making a bar graph in Excel. So this particular data set I actually got from one of my honors experiments, which is using fluorescence to determine the amount of phagocytosis of these fluorescent E. coli bioparticles. That's not particularly important, but as you can see, I have a bunch of treatment conditions, and for each condition, I have 4 replicates, each with a fluorescence reading, which are the numbers seen here. Okay, so I'm going to be going through some data processing and some stats, which definitely has some helpful Excel tricks, but if you want to just skip to the figure formatting, you can go ahead and skip to 8 minutes and 35 seconds. Okay, so in processing this data, the first thing I want to do is I want to subtract the background fluorescence from all the other values. So to do this, I take my no cell control, which is the background, and I'm going to average this by doing equals average and selecting the numbers. And with that, I am going to be subtracting the average background from each of my values. So to do this, I am going to take the value I want to subtract from. I'm going to do equal and then click the value that I want to subtract from. Then I'm going to go minus and then this mean background here. Now I could do this for every single one, but that would be very time consuming. So what I'm going to do is I'm going to put a $sign in front of the F, and that will keep my selection on the F, and then a $sign in front of the 14, keeping that row 14. And then I can take the little green box in the corner here, drag it all the way down, and as you can see, the number I'm subtracting from is always subtracting from F14, which is the mean fluorescence for the no cell control. I can also drag this all the way across, and it will do the exact same thing, always keeping that subtraction of that column F, row 14, no cell control background. Okay, so the next thing I want to do is calculate the fold change between my treatments and the no treatment control to see what the effect is of each of the treatments. To do this, I want to divide my treatment condition by the no treatment control. So what I'm going to do here is first take the average of my no treatment control, and then I'll divide each of my values by that mean. So I go equals average, select my values, and then click enter. Okay, so then I want to take the fold change for each of my values, even the replicates of the no treatment control, so that I can get some error bars in my figure. And so what I'm going to do first is press equal, select my data minus that background already, and then divide it by the mean no treatment control. Again, to save time, I'm going to select the $sign function to always be dividing by that column F, row 18, mean of the no treatment control. And then I can drag through my values, and again, you can see that it's keeping the number we're dividing constant. And then we can do the same thing across the columns. Again, you can just see it's always keeping that dividing number the same. Okay, so now what we want to plot is the mean plus standard deviation. So to calculate the mean, we do equals average, brackets, select our values, and then enter. And if we select that little green box in the corner there, drag it down, because there are no dollar signs, it will take the average of each row as we go across. Similarly, to do standard deviation, we use the function stdev, bracket, select our values, enter, and again, we can drag that all the way down. Okay, so I want to do some statistics on this data to see if we can see a statistical significant difference between our treatment groups. So to do that, I'm going to use a t-test, and I specifically want to see if my treatment is different than my no treatment control. So to do that, I'm going to use the function t.test, bracket, and then you select an array of numbers. So the first set of numbers you want to compare. In this case, I want to compare everything to the no treatment control to see if my treatments are making the difference in phagocytosis. So I'm going to select all of the no treatment control values. Next, I put a comma and I select my treatment values. And next I put a comma and I can select either a one-tail or two-tailed t-test. One-tail is if you're checking only if it's higher than your treatment. I want to see if it's higher or lower than my treatment, so I select two-tailed 2. Next comma, it gives you an option to do paired number 1, which you would use if you're using the same group over time. So for instance, the same plant over time. Mine are different groups with different conditions, so it's unpaired, and I'm assuming the variance between groups isn't necessarily equal, so I'm using a heterostatistic test and selecting 3, enter. So instead of typing this over and over again, I'm going to use the $ trick. In this case, because I'm always comparing to the no treatment control, in the array, I'm going to put the $ to select from B32 to E32 in the first array. And then I can just drag all the way down. And as you can see, every time it's comparing the treatment to the first set of no treatment control groups. This is important because if our t-test value is under 0.05, we can declare that our treatment condition had a different amount of phagocytosis than our no treatment control. Okay, now on to graphing. Here I want to select my different treatment groups as my bars, and then I will be plotting the mean of those bars. So I would hold Command on a Mac or Control on a PC, and then select the mean column corresponding to those values. Once that's selected, I go into Insert, 2D bar graph. Here I just am going to make it a little bigger. Bring it to the side. And for CSC, we don't want any grid lines, so I'll delete that. And we don't want a title on the top, so I'll delete that. And now we want to add some chart elements. So on the very top, you can see I'm under chart design, and then you can go to the left with that little bar symbol. I know I want to add horizontal and a vertical axis title. So here, horizontal, I'll put my treatments. And then for vertical, I would put phagocytosis because that's the experiment, and then in brackets fold change, but for simplicity I'll just put fold change and then relative to the no treatment control. Okay, so next I want to add my error bars. So to do that, I'll go to chart design at the top, add chart element, and select error bars. We want to use our own calculated standard deviations. So we're going to hit more error bar options. We get this window popping up at the side. We're going to hit custom. And then we are going to specify our values. There's a little check box here to select values from your sheet. We are going to select our standard deviations as specified by their specific row. Hit enter, and then we can do the same thing for the negative error bar. Hit enter, and when we click OK, it's going to put in our values. As a little check to ensure the right standard deviations are in the right rows, you can kind of just look at the big error bars and see if they're corresponding with the bigger standard deviations, which as you can see, they do here. So now for a little more formatting, if we want to adjust our axis titles, we can just click on it and edit it how you would normally edit text. So I'm making it black font and a little bit bigger in size. And you can do the same for the treatment titles and the vertical axis. Then I also want to make the line at the base of the figure black. So I click on it, click the paint bucket symbol, and then select solid line. Same thing for the vertical axis. You can see in the bar section, you can edit the actual bullet numbers. I just want to make the axis line black, so I go back to the paint can, select solid line, and then it is perfectly formatted there. Now to edit the bars themselves, you can click on them, go to that paint can symbol, and then just change the fill color. If you want to do them individually, you can double click, change it, change the fill color. If you do white, you want to put outline so you can see it, but you don't have to do that for the rest of them. Another thing you can do, if you want the standard deviation to just be positive, you can double click on it and then just select the plus. I think that looks a little bit nicer. Okay, so back to the colors. You usually want to keep it grayscale. So just select colors within the grayscale. Just for fun, I'll use some different colors. So I'll make one purple. I'll go blue, green, orange, and red. I'll do the last two grayscale. You do want to keep it grayscale for standard CSV formatting. And then the last thing you want to do, you want to make sure that there's no border around the figure. So to get rid of that, you can just click on the figure, go to that paint bucket icon, select no border, and then you're done. Then you can just copy it and paste it into a Word document or PowerPoint, whatever, wherever you're putting the figure. And then you want to make a figure caption. The figure caption goes below, and you kind of want to just describe the figure so that somebody looking at it could understand what it is and everything the graph is showing. So yeah, here's a little example, but there you have our beautiful figure. And in the next video, we'll go on to creating the standard curve and table formatting.